Hi friends, today we are going to discuss about the factor about high frequency recombination and it is a question of 2017 December booklet code day and question number 113 and the question carrying 4 marks. And before going to discussion, we have already mentioned here that it is a part of bacterial genetics. And before going to start this problem on numerical part, uh, we have to know one thing: what is F factor? F factor is nothing but the fertility factor and was first discovered by Ether Lederberg, and this factor was discovered by Ether Lederberg and also called, this factor is also known as, this F factor is also known as sex factor. And I am discussing this matter as because this kind of question is very common in CSIR net. In the last two session 2019 June and 18 December does not carrying such kind of question. So 2019 December it is a very important for this kind of question in CBT mode. So please keep concentrate such kind of question. These are not so difficult, but if you clear through your basic knowledges about this matter, you can easily solve this kind of problem. So now again we come to the matter, and this factor is known as sex factor, and in E. coli or the F sex factor it is also known as that, the F sex factor. F sex factor, this is also known as the F sex factor, and they also known as F plasmid. Various kinds of names are there we can use. So during this session we found that F minus we have already discussed here, F plus and F minus, such kind of term we also found in this case. When a F plus conjugate met with an F minus conjugate, such as suppose F plus is crossing with an F minus conjugate, then the result where they meet, the result 2F plus conjugate, and the conjugates are both are capable of transmitting the plasmid to other F minus cell, and they both capable of carrying this plasmid to another class of during conjugation, and the F plasmid belong to the class of conjugative plasmid. They are also known as conjugative plasmid. So this kind of basic knowledge we also need before we start the discussion. But before start the numerical problem, I want to say one thing, that such kind of knowledge, basic knowledge is necessary, because this kind of question may create a fear in our mind in the exam session in the hall. So please don't be panic such kind of question, such kind of question, because this kind of question not needed such kind of knowledge, but we have to clear this, because without clearing it we cannot easily succeed. That what are they, if a new pioneer found this kind of question or question pattern, they easily get panic through it. So we have to know such kind of information before it. So another term we found in this year is HFR strain. HFR strain is nothing but the high frequency recombination strain. High frequency recombination strain. So these two terms are very necessary, because these two terms we have found that these two terms are present in the genetical problem, as we found in this. A high frequency recombination cell or strain is a bacterium in which conjugative plasmid integrated to the chromosomal DNA, and the integration of the plasmid into the cell of chromosome is through homologous recombination. The HFR plasmid just doing nothing, they just carrying the factor, and integrated into the chromosomal DNA of another chromosome. So and it is nothing but the recombination, and this is HFR carrying some kind of DNA, and they add this to the another chromosomal part they add, and this kind of recombination is nothing but the homologous recombination. So now we can start the problem, because now we are clear from this terminology what is homologous HFR strain, what do you mean by F plus or F minus strain. So in this numerical problem, we found that they have given a chart, the three HFR strain they have given to us: HFR1, HFR2 and HFR3. Three kind of gene they have showed to us, and the genes and time they have showed. One they have told about the genes, and another is time. In each case, genes and times. And lastly, they have in every case they have discussed here the genes and times, and in every case they showed us a time, because during the recombination, the transformation of gene from one part of the chromosome carried by the plasmid to the another chromosome, when recombination occur, the time shows that how many distance they are carrying. Like in this first part, if we found that E plus is 6, E plus, they have arranged a pattern like E plus, F plus, C plus, D plus and B plus, and in the HFR2 they have found that B plus, D plus, C plus, F plus and G plus, and in the last pattern they have showed that D plus, C plus, F plus, E plus and G plus, and every time they have showed a time, like in E plus first HFR strain H 24, 34, 49 and 54, and in the second HFR strain they have showed 1, 6, 21, 31 and 63, and in the last 4, 19, 29, 47 and 61. Now they have given that the pattern order of gene, we have to calculate the order of gene and the distance between the gene. But how could you discuss about the distance between the two genes? The distance between the two genes calculation is very easy. Distance between two gene calculation is very easy. You can easily calculate in such a pattern, like E plus in the first pattern and F plus carrying 24 and E plus carrying 26. If we minus F 24 from 6, so 18 is the E plus F plus distance is nothing but 18. So in such a way you can calculate in every distance between them, like here is 18, here is 10, here is 15, here is 5. The similar way you can find that 5, 15, 10 and 32. The similar way in the HFR strain third, 15, 10, 12 and 20. So In such a way you can easily calculate the distance. But I want to show you in this trend that the distance between the two genes, like we can say that B plus D plus in the HFR strain shows the distance of 5 minutes. In the similar way, in HFR strain 2, the distance between B plus and D plus is the same. So the difference between the two genes, or the distance between the two genes in the 3 HFR strain are same. The first, second and third, they always remain the same distance. So if we calculate in one strain, we can easily calculate the another strain easily. So we don't have to calculate as many as time. So they have given the distance option. Now we can see the distance option. They have show the distance in the option, that is F and G. We can calculate in between F and G, where we can find that F and G. Firstly, F and G, F and G is 32, and another F and G here we can found that only in the HFR2 strain 2 we can found that F and G is 32 distance, and F and B they want to calculate 30. The distance between F and B we can found here somewhere, where the distance between F and B, B and D, F, C, F, B, like we can found like F is 24, F is 24, and we can see that B is 54. So the distance is in between them is 30. So F and B distance is 30. You can see here in the HFR strain 2 that B is 1 and F is 31. So the distance remains same, that is 30. So in the first option, C is right. They have given a option that F and G distance is 32 minutes and between F and B distance is 30. So option number 3 is the correct option. So we can say correct. So from this combination we can easily say that number 3 and number 4 is not correct. So we can easily discard it from these options, and there are two options in our hand. Another is 1 and 2, and in the another page we can, they has given that C and E distance is 28. Now we can calculate C and E, from where we can get that C and E. We can see C we found in the first option, C is equal to 34 and E is equal to 41. So the no 41 not, it is 47, sorry. So the distance between them, that is 18 here. We have miscalculated here due to wrong writing process. So E is 47 and C is 21. So we can easily say that the distance between them is 21. So we can easily, or 6 is E and C is 34, sorry, E is 6 and C is 34. So you can easily say that the distance between them is 28. And in the similar way we can found if C and E in the same HFR. No, there is no same recombination. C19 we have found in the C19 and E47. So here the distance is same. So we can easily found that 28 is the distance. So C distance is right, that is 28 minutes. And another distance they have given, that is B and C. B and C we can found that C is 34 if we found, and B is 54. C 34 and B is 54. So the distance between them is 30. We can easily calculate from here. And if another here B and C, there is here in the HFR strain 2 we can found that B is 1 and C is 21. So distance between them is 20. So the distance between B and T is 20. So the distance of, as they have given that B and C distance is 20. So the two options are already right. So C and D both are correct. So C and D are present only in the option number 1. So B and C is also, so the option 2 is also discarded. So only one option, but we have to calculate the gene order. Before we going to the gene order, they have given us the two gene order. In the gene order, first they given E, G, B, D, C and F, and in the second option they have given the F, G, B, D, C and E. We want to show you one thing before we start the calculation, that the middle part like GBDC and GBDC are same, but the difference between them, in the first A option E has the gene recombination started with EG and the second the FG, but we have to calculate the distance which is smaller, that is calculate they are closely related, like we have to discuss E and G the distance, and we found that in E is 6. You can found that E is 6 and G is, we can found that G is 63. E is 6 and G is, we can found here G, where is G? E and G in the same order we can found here 47 and 61. So we can easily say E is 47 and G is 61. So distance between them is 14. From here we also found that if the two genes are present in the same frequency, then we have to calculate there from first. If they are in different frequency, they have show different results. Like here in the first frequency is 6, but in the second frequency there is 63. So the distance we calculate that is wrong. So we have to always first find if the frequency remain in the same frequency, if the genes are present in the same frequency, so we can calculate easily. So E and G distance is 14, but we have to calculate F and G. The F and G is the distance between them. We have to calculate that F and G. F and G, in F that is 31 and G is 63. So the distance between them is 32. So in between the, and it is present in the HFR strain 2, and we found that E and G, it is found in HFR strain 3. So we can easily say that the distance between E and G are similar. So this combination should be more perfect than the B. And lastly, the distance between C and F, and we have to find that the distance between C and F, we have to find between the distance C and F and C and E, what should be the distance of C and F gene and what should be the distance of C and E gene. So first we have to calculate what is, if C and F are present in the same combination, we can found here that the C and F found in the HFR frequency, F is 24 and C is 34. So distance between them is 10. In HFR frequency we have found that C is and F frequency is found in the HFR strain, that is 10, but in C and E, if we found that C and E, we found in the HFR strain C and E, we found in the HFR3 where C is 19 and E is 47. So distance between them is 28. So the distance is more closer in case of C and F. So this option is right. This option we will not take. This is more appropriate. So C and E is not correct, just like before. So C and F is the correct combination. The middle part remains same in these two cases. So the option A is the right recombination. So we can easily say that as we have already got that if we have not much less time, not much time in the exam room or exam hall, we can easily find that C and D is not present in anywhere in these three options. So this is the appropriate answer, but we have to calculate here for the students what should be the correct answer. So question option number 1 should be the correct answer. So option 1 is the right answer. So now today we have discussed here a bacterial genetics part, where we have discussed that bacterial genetics part where we have discussed about the bacterial genetics part where we have discussed about the plasmid recombination HFR and F plus plasmid and F minus plasmid, and we have very small discussion we have done there due to, and also we have mentioned about how to calculate the distance, and arrangement of genes can be done. So this kind of questions are very important for 2019 December net, because from last 1 year this kind of question is absent. So please keep concentrate such kind of question, and this is a very easy question. If you concentrate on it, you can easily done within 2 minutes to 3 minutes maximum. So thank you guys for watching this video. Please click the like button for this video and don't forget to subscribe my channel. Thank you very much.